Okay, good afternoon, everyone. Thank you for attending today's Applied Plant Sciences seminar. Today, Logan Spicer will be presenting on investigating novel de novo meristem induction through a direct delivery gene editing technique in greenhouse-grown apple and grape. Logan is currently an APS master's student working with Dr. Matt Clark and Dr. Jim Luby. Logan earned a bachelor's degree in biological science with a minor in chemistry from Rowan University. And previously, Logan has worked in the aquatic biology undergrad lab at Rowan University and has done ecological surveys work in Puerto Rican forests. And a fun fact about Logan is that he is good at swimming. Go ahead and take it away, Logan.
All right, thanks, Lita. So with that, I guess I'll just jump right in. So Malus domestica, or the domesticated apple, is a perennial woody fruit crop, part of the Rosaceae family, and annually there's about 75 million tons of fresh fruit produced worldwide, and this is from the 2020/2021 fiscal term. Some types of apples would be dessert/fresh apples that you can buy at the store. You also have your cider apples, your apples for baking, and you have your crab apples, which are mainly just for pollination. Popular cultivars would be Honeycrisp apples, which were developed at the U here, Granny Smith apples, and Red Delicious apples, all of which can be bought at your local grocery store. So moving on to grape now, or Vitis vinifera, the common domesticated grape. It is also a perennial woody fruit crop. It's part of the Vitaceae family, and there are about 25 million tons of fresh fruit produced annually worldwide of grapes. So some types of grapes, you have your wine grapes, your table grapes, which are the kind that you buy at the store and eat. And you have your grapes that are dehydrated, like raisins and things like that. Some cold hardy varieties, I kind of wanted to just mention a couple names. All these varieties were developed at the university here, and they include Frontenac, Itasca, and Marquette. So a quick summary on the genetics of these crops. So apples have a total of 17 chromosome pairs. They are diploid. The estimated genome size is between 650 and 750 megabases, and they are self-incompatible. So self-incompatible essentially just means that the apple tree cannot pollinate itself. So if you were to go to Lowe's and buy an apple tree, plant it in your backyard, you would then need a second tree, either in your backyard or in close vicinity, that can then pollinate your first tree to get fruit. For grape, number of chromosome pairs is 19. It is also a diploid, and the estimated genome size is slightly smaller at about 500 megabases. And these are self-pollinating. So let's get into the breeding aspect of apple and grape. So some breeding challenges for these crops would be that they're highly heterozygous. They do not breed true from seed. And this essentially just means that if you were to go to the grocery store and buy a Honeycrisp apple, take the seeds out and grow a tree from seed, the tree that grew will not be the same as, it will not bear fruit the same as the one that you bought at the store. Commercially, these are asexually propagated by grafting onto rootstocks, and the juvenile phase is about 3 to 8 years. So for grape, it takes about 3 to 5 years to produce fruit, and for apple, it takes about 5 to 8 years. So the development timeline for cultivars for apple and grape is a very lengthy process. It takes decades to produce commercially viable genotypes. And some examples of this would be in Honeycrisp, the original cross was designated in 1974, and it was released in 1991. And Marquette, a cold hardy grape variety developed at the U, was crossed in 1986 and released in 2006. And these both took about 17 years to go from cross to being commercially available to growers. So about 1 out of every 14,000 cross receives a name and is introduced to the public. So this stat is actually for the apple breeding program, but I'm sure that is also a similar number with the grape breeding program. So some biotech techniques to reduce the breeding time usually involve genetic markers, genetic mapping, whole genome sequencing, or genetic transformation systems. So we're going to continue with talking about genetic transformation systems. So this is essentially the production of genetically engineered plants. And a genetically engineered plant is a plant with genetic material that has been altered through means that do not occur naturally by mating or by natural recombination. And this includes gene editing methods to further break it down. So what is gene editing? Well, gene editing is essentially targeted manipulations. It is the precise use of DNA or RNA guides to delete, modify, replace, or insert DNA into another organism. So types of gene editing approaches. There are many types of approaches to gene editing, but these are kind of the big three in plant breeding or with plant science. So you have your zinc finger nuclease, you have talons, and of course you have your CRISPR-Cas9, which is the most popular way right now. So CRISPR-Cas was first identified in E. coli as an antiviral defense mechanism in bacteria, and it was first used for its editing applications in mammalian cells in 2012. There are 6 major types of CRISPR systems. The most common for gene editing in plants is CRISPR-Cas9, which is a type 2 system. And the CRISPR systems are designated by their function and their genes present. So what is CRISPR? I'm sure you've all seen this acronym thrown around, but like what actually is CRISPR and what does it stand for? Well, CRISPR stands for clustered, regularly interspaced, short, palindromic repeats. And the whole point of this acronym is just a way that you can identify your CRISPR in a kind of like you can identify the sequence in a gene. So to further break down what CRISPR-Cas9 is, you always see this phrase CRISPR-Cas9. So we already broke down what the acronym CRISPR stands for as a way to identify CRISPR in sequence, but what does the Cas stand for? The Cas is essentially just the nuclease protein that cleaves your nucleotides, and the 9 refers to the associated protein. So Cas9, the ninth nuclease. How does this work? So in purple, we have our Cas9 protein nuclease, which does our cutting. In blue, we have our genomic DNA template. And in green, we have our guide RNA that guides our cut. And in the orange here, we have our CRISPR RNA, which is the RNA in the protein, and we have our tracer RNA, which holds the CRISPR RNA in place, kind of. So essentially, the tracer RNA and the CRISPR RNA form a complex that bind to the Cas9 nuclease and kind of direct it to the target sequence, right? And then you have your PAM in the red here, which essentially just stops your cut. And what a PAM is, it's an acronym short for protospacer adjacent motif, which is just a small 2 to 6 base pair sequence following another 2 to 4 base pairs from the cut site of the targeted DNA region. And this can be identified in sequence as NGG. So any nucleotide can be N followed by 2 Gs in the sequence. And this whole process generates a double strand break. So why use CRISPR-Cas? So it has the ability to precisely manipulate targeted genome sequence. It is possible to edit or knock out a specific gene. It has low time and resource limitations. And it is predictable, cheap, and fast. So Maher et al. 2019, I will be referring to this paper quite a lot. So Maher et al. Titled Plant Gene Editing through De Novo Induction of Meristems developed a gene editing approach in soil-grown tobacco plants, resulting in gene editing techniques alternative to tissue culture-based methods. So what is de novo induction of meristems? What does this mean? Well, in Latin, de novo means from the new. So inducing new meristematic shoots is what it essentially means. So creating new meristems by delivering developmental regulators and gene editing reagents to semantic cells, which are just undifferentiated cells. New meristems that form into shoots with targeted gene edits can then be passed on to the next generation. And we took methods and techniques from this paper to develop and test our own experiments in our target crops. This is one of the only papers that's kind of conducted experiments on soil-grown plants with gene editing. So moving on, I kind of mentioned developmental regulators, but like, what are developmental regulators or DRs? Well, DRs are genes that stimulate the production of cytokinins, and cytokinins are hormones that are responsible for cell division and development in plants. So essentially what happens here is the developmental regulator genes stimulate the production of cytokinins in the plant that induce new meristem growth that then forms into shoots with edited tissue. And we use the developmental regulator isopentenyl transferase, or IPT for short. And this was identified from Maher et al. As just the overall best developmental regulator that he tested out. It's the best for growth and the best for these kinds of experiments is what he found. So some vectors here. Again, from Maher et al., we followed their approach in constructing a modular backbone of our gene editing reagents. And the modular backbone can then be cloned and transformed into a tDNA vector. And our modular backbone is made up of our Cas9, which does our cutting, our guide RNA, which guides our cut, our developmental regulators, which produces meristematic growth, and luciferase, or Luce for short, which is our bioluminescent reporter that we use to check the efficiency of our gene edits. And our tDNA vector is a Gemini viral replicon, or GVR for short. It is agrobacterium mediated, and the agrobacterium, by agrobacterium tumefaciens, strain GV3101. So again, I mentioned agrobacterium, but what is it? Essentially, it's an intermediary to transfer DNA to a target plant, and was originally found as a soil phytopathogen that naturally infects plant wounds, and it causes crown gall disease by transferring DNA from the pathogen cells into the host cells. And the picture on the right here, you can see this very tall tree has this large tumor or callus forming on, kind of just engulfing the trunk of it. So this is an example of crown gall disease by Agrobacterium tumefaciens. So again, from Maher et al., we followed their procedure where they designed their modular backbone. So we have our modular backbone up at the top left up here. And this is made up of the reagents, of the backbone using kits. So similar to like QIAGEN kits that you'd use for like DNA extractions, there are kits where you can use to make your CRISPR, your guides, and like any reporter genes that you may have. And then once the reagents and the genes have been kind of completed and made up in lab, they are cloned into plasmids. And what plasmids are, they are small circular DNA strands that replicate independently from the chromosomes of the organism. And then the plasmids are then assembled into tDNA vectors for agrobacterium transformations through Golden Gate assembly. So moving on to our experiment and our treatment groups. we designed 4 modular backbones to be assembled into tDNA. The treatment groups are made up of our Cas9, our guide RNA, our developmental regulator, and our bioluminescent reporter. We made 2 different treatment tDNAs. So we assembled 2 different tDNAs that were assembled, one with the guide for our apple target gene and the other for our grape target gene. So the one, excuse me, the only difference is that they have different guides, but they do exactly the same thing. The first control group that we have is assembled of just our luciferase, which is our bioluminescent reporter, and our IPT, which is our developmental regulator. And lastly, our second control group only has our luciferase plasmid inside of it. So this is what we expect to see from each experiment group. So in our treatment group, since we have our cast, we have our guide, we have our developmental regulator and our bioluminescent reporter, we expect to knock out our target gene to see increased meristematic growth and to produce bioluminescent shoots under UV light. With our first control group that only has our bioluminescent reporter and our developmental regulator, we do not expect to see a knockout, but we do expect to see increased meristematic growth and bioluminescent shoots under UV light. And lastly, for our second control group that only has our bioluminescent reporter transformed in it, we do not expect to see a knockout and we do not expect increase in meristematic growth, but we do expect bioluminescence in our shoots under UV light. So direct delivery preparations. This is how we prepare injection solutions for our experiments. So starting with our stock tDNA cells for our experiment groups on the left over here, they are stored in -80 freezers, and they are in glycerin stocks to kind of keep them fresh or dormant. We then use a pipette tip to scrape cells directly onto Petri dishes with LB medium. And LB medium is essentially just media that is good for cell growth. We incubate our Petri dishes for about 48 hours or 2 days. And after incubation, we take a single colony from each of our Petri dishes and we stir them into a beaker with activation media. And again, activation media is liquid media that is good for cell growth, so our cells can grow and culture. And it's also good for activation/selection of our gene editing reagents. So we incubate our activation media with our single colony in it for 20 to 24 hours, depending on how high you want your cell concentration to be. And then after incubation, we pellet down the our liquids into cell pellets using a centrifuge. And then we resuspend the pelleted cells in infiltration media. And infiltration media is essentially the cell suspension buffer to keep the cells alive long enough for our injections later on. And we took this method from, of course, Mayer et al. So direct delivery. This is the method that they developed in Maher et al. To avoid tissue culture. Like I said, it's called direct delivery. So by taking a whole plant and removing existing meristems, editing reagents can then be injected into the delivery site from the removed meristem. New meristem formation begins producing a new shoot with fixed modifications that can segregate in offspring. So we're using this method as a jumping off point kind of for our experiments. But of course, we have some modifications to it just because of the target plants that we are using. So a summary so far of what we've talked about. So here are kind of just like the big takeaways from the presentation up until now. Apple and grape are economically important fruit crops worldwide. They have a long life cycle. Takes a while for them to bear fruit, and they have long variety release times. Remember, it takes decades to produce commercially viable genotypes. We talked about CRISPR. So CRISPR-Cas9 potentially can reduce cultivar development time with precise genome editing. And we are applying methods developed by Maher et al. To our own target crops to kind of see if we can shorten this development time. So significance here. So using methods from Maher et al., we hope to bypass limitations of traditional breeding with more predictable and precise outcomes. We want to avoid tissue culture. Tissue culture is difficult in woody species, especially in apple. It is possible in both apple and grape. In fact, I have several tissue culture grape plants growing in our lab, but it is still an arduous process. It takes a lot of time, it's very laborious. So we're taking, what we want to do is, so we know that gene editing in soil grown apple and grape vine is very understudied. And we kind of want to contribute to the investigation of gene editing efficacy in soil grown plants. And I personally find this aspect very exciting about my research. We're kind of on the forefront of this. So potential applications of using CRISPR-Cas9 in our soil grown apple and grape plants. So potentially we could reduce time costs in apple and grape breeding programs. This can be used in elite lines to quickly implement a trait. We can create plants to investigate gene function. We can change a single trait in an established variety without changing several other traits, which can normally take about 15 years using traditional breeding methods. And this is especially helpful if you want to change traits in cultivars with long commercial lives already. So moving on, I kind of wanted to drive home the potential application of changing a single trait in an established cultivar without changing a bunch of other traits. So this figure over here was created by Seth Wannemuller for the apple breeding program to kind of illustrate the apple breeding process at the university here. So you kind of start with your crossing stage, and after about a year, you then move these into your greenhouse growth stages. After a couple of years, you can start your stage 1 seedling trials when they start to bear fruit. And then after this, you go into your stage 2 clonal trials. And then after year 7 to 16, you then start your stage 3 trials, which are just commercial trials for growers and things like that. And then after about 15 and 25 years, you may find your golden tree, which would then become a commercial variety. So an example of this of our application would be Red Delicious apples. They have been on the market for about 50 years, or longer than 50 years actually. But what if we had a candidate gene that we wanted to take out to update this cultivar? We can use our development methods to knock out a single gene in this established variety. And then to test this, instead of starting at the crossing stage here, we'd actually be able to start at this golden stage and kind of just jump past all these other steps and go right into stage 3 growth testing. And then from here, if the new treat performs well and growers decide to pick this up, and the growers decide to pick it up, a new cultivar, the old cultivar can now be updated, and Red Delicious apples there's now a new, more updated Red Delicious apple variety on the market. So what we know from the literature in apple, we know that CRISPR-Cas9 works in apple. They've done studies on this in rootstock varieties and as well in commercial varieties. Most of these experiments, though, have been in vitro. So almost all of these experiments have essentially just been done using tissue culture methods via like callus cultures and things like that. But little has actually been done on soil grown apple seedlings or plants. And I kind of wanted to mention Arctic Apple in this slide. So what they did at Arctic Apple is not technically gene editing. It is genetic transformation. They didn't really edit out a gene for their product. They silenced the gene, and I kind of decided that this was close enough and can illustrate my point well enough. So essentially what they did was they found their candidate gene in apple for browning or oxidative response, and they silenced this gene. And now they have new apples for Golden Delicious and Granny Smith that do not brown. So the way they kind of market this, you can see in this picture here, is that they have their apple slices and they cut them all up, they throw them in the bag where it's very visible of the inside contents, and you can see that the apples do not brown. So this is like a pretty cool application of what we could potentially do in the future with apples. So moving on to what we know in grape. So we actually know a lot more about CRISPR-Cas9 gene editing in Grapevine. They're kind of at the point now where they're not just testing to see if it works, but they're testing the efficiencies of using CRISPR in Grapevine to actually see how far they can go and what else they can learn. And again, all of these studies have pretty much been in vitro or using embryonic calli. They're doing studies where they try to increase powdery mildew resistance by knocking out a gene or inserting genes. And again, I have reiterated this so many times, but experiments in soil grown grapevine is like non-existent for the most part. But in the paper Maher et al., which I keep referring to as well, they did experimentation in grapevine. However, this publication mostly discusses the research in other plant species. Where he does mention his experiments in grapevine, they are done in tissue culture. However, when speaking with the authors in person, we actually found out that he did do experiments testing his direct delivery method in soil grown grapevine. And we found that this is actually possible to do. So you want to take more of an applied approach for our next experiments. So gathering what we know and what we don't know, what's available and what's not available information-wise, we formulated these objectives. So our objective with Apple is to determine the feasibility and efficiency of de novo meristem induction for gene editing in soil-grown apple seedlings. And essentially, this is just a proof of concept. In grape, we want to implement a targeted knockout of a candidate gene identified for phylloxera resistance in a cold, hardy grape hybrid. And this is more of our applied study. We know this works in grape, in soil-grown grapevine. So now we actually want to try to take out a candidate gene. So continuing with apple, I'm not going to repeat the objective since I just said it, but essentially we want to develop and test methods for gene editing in soil-grown apple seedlings. And this is what our hypothesis is. Direct delivery of a gene editing construct to wounded nodes will result in the formation of new bioluminescent shoots with a targeted gene knockout. And our target gene here is called phytoene desaturase, which I'll refer to as PDS, and is a commonly used reporter gene. If a company is deciding to test the efficiency of gene editing using CRISPR-Cas9 in their plant species or their target species, they almost always use a reporter gene, which is usually phytoene desaturase, and I'll tell you why. It is part of the carotenoid biosynthesis pathway. So essentially it's part of, it has something to do with photosynthesis and pigmentation. So the mutagenesis of this gene results in albino and dwarf phenotypes. So down here, in this little picture that I kind of diagrammed here, you start with your green leaf, right? You take out your PDS gene, and then hopefully you get a nice albino and white phenotype. So the plant material that we're using are apple seedlings. This is just a fast way to get a lot of plant material. So these were propagated from seed. They were grown in the greenhouse. They were open pollinated seedlings from 5 cultivars: Honeycrisp, Regent, Frostbite, Miniesca, and Snowsweet. Total plant material that I have is about 300 seedlings from each cultivar. I also have a bunch of seeds stored away in case I need more plant material for whatever reason. And here's just a figure of early on my apple seedlings. They're not this small anymore, they were much bigger. So let's get into our direct delivery. So very similar to what Michael did with some exceptions, we take our whole plant and we prune the top. So we prune the top of our plant for a couple reasons. One, because sometimes the plants are very tall, so it's easier to prune them down to make this process easier. But it's also good to prune them down to kind of induce vegetative growth and to also disrupt apical dominance so we get bud breaking on our axillary nodes where we want our meristems to form so we can get our targeted knockouts. So we prune our top, we designate our nodes, and we perform our apple bud slit kind of. So we have our node kind of identified here with the leaf coming out of it. We take the leaf off and we perform a horizontal slit across the face of our node. And it kind of looks like this. This is a little bit over exaggerated of a cut because I wanted the cut to be visible to show you all. So then from here, we defoliate and we wound our plant with the slit, and then we inject into that node. So after injections, we cull all new growth for 20 days after our initial injection, and this includes our injection nodes. And we do this because we want to make sure that edited shoots form, and we completely remove any latent buds that may be inside that node. And then after 20 days, we let the injection sites grow, but we still continue to cull all other growth at non-injection sites on the plant. So after letting the plants grow and form full leaves, we want to check the gene editing efficiency. So what we're looking for is a complete knockout. And we can do this visually actually because of our PDS gene and how if it is successfully knocked out, it will produce a white phenotype or a dwarf growth phenotype. It is also possible to kill the plant, but in my opinion, this isn't a really good indicator of the efficiency of our experiment, but that is also something that we need to look out for. So if we do see white, or if we see some dwarfing or anything abnormal, we then proceed to our Luciferase assay. So as I mentioned before, when we were constructing our tDNA vectors, if you noticed, all of our tDNA vectors for every experiment and control group have luciferase in them. So since they all have luciferase in them, we can then treat these leaves with luciferin. And when luciferin and luciferase are together, you can observe the presence of bioluminescence under UV light. So again, like this cartoon kind of illustrates, you have your green leaf, which should have your luciferase in it if it was part of any of my experiments. You put your luciferin in, it does some sort of chemical reaction, and then under UV light, you get this nice bioluminescent blue color. So just to summarize our experiments, we prepare direct delivery, we perform our injections, we cull all growth for 20 days, and then after 20 days, we let our injection nodes grow out, and then we check the efficiency of our edits. And this is the same pipeline we use in Grapevine for Grapevine experimentation. And currently, here are some current experiments that we are currently conducting. So we're doing some experiments on vegetative tissue versus lignified tissue, which essentially is just testing this experiment in different stages of growth. So we would do experiments in younger plants about 3 months old where they're very soft and very vegetative. We also do experiments in plants that are about 6 months old, which are still green and vegetative, but they're starting to form more lignified tissue around their stems. And then we do this also in more mature plants at about 9 months old, where they're pretty much completely lignified at this point and very hard to inject into. We're also doing experiments where we wrap parafilm around our injection sites. And this is just to kind of retain moisture so they don't dry out, retain humidity, and to kind of try to keep our cell suspension in contact with our node. We also did experiments by keeping leaves on at injection nodes, but cutting all of their leaves off. We also experimented with different degrees of node cutting. There's also some other future experiments that I have floating around in my mind that include using higher cell concentrations. So currently we use cell concentrations of about 0.2, of an OD about 0.2, which is just the density of your cells in your solution. And I kind of want to try upping this to maybe like 0.5, 0.75, or maybe 1. But we're using 0.2 currently because that is what Michael was, or what Meyer et al. Was using in their methods. And we're also thinking about maybe doing experiments where we keep all the leaves on the entire plant and we cut off just the injection nodes and inject like that. So future directions for Apple. So with a successful knockout, we can then test in commercially available clonal varieties. We can perform knockouts on candidate genes, fine-tune procedure efficiency. If this doesn't work, if we don't get knockouts, we can just perform more experiments and just keep fine-tuning our procedure efficiency. So now getting into Grape, I will reiterate our objective, which is to implement a targeted knockout of a candidate gene identified for phylloxera resistance in a cold hardy grape hybrid, while further developing and testing methods for gene editing in soil grown grapevine. And this is, again, the more applied study. So our hypothesis in grape is very similar to our hypothesis in apple. So we expect direct delivery of a gene editing construct to wounded nodes will result in the formation of new bioluminescent shoots with a targeted knockout of our candidate gene. So I mentioned phylloxera quite a lot with grape. And what phylloxera is, or grapevine phylloxera, is a major insect pest that is responsible for a lot of worldwide economic damage seen in vineyards, in commercial grapevines, I should say. So this is found in most grape growing countries, actually, like around the world, except for a couple like New Zealand and China come to mind. The native range is hypothesized to be North America. And it's kind of tragic how, I guess, in the 19th century, Americans kind of brought American Vitis vinifera plants over to Europe to develop powdery mildew resistance, and they ended up bringing this whole other pest, and it just decimated the European vineyards in the 19th century, and it's a total mess. So what does phylloxera actually do? Well, part of their life cycle is they form galls on leaves and roots. So on leaves, it diminishes photosynthesis, and it kind of just doesn't allow the plant to sustain itself and make its own energy. In roots, it forms the galls, and it kind of just systematically collapses the rooting system. To mitigate this, there are rootstocks available and better vineyard practices that you can use, but they only mitigate the damage. They don't really stop it. So I kind of wanted to highlight here what the galls actually look like. So in the top picture, the galls are not the white part, that is perlite. The galls are actually the lighter yellowish brown, slightly swollen roots. And on the bottom picture, you can see in a grapevine, the galls form on the underside of the leaves here. So Dr. Lu Yin, she is responsible for identifying our candidate gene for foliar phylloxera resistance, and she is the reason why this project is possible. So thank you, Lu, if you were watching. She used a biparental F1 population of cold hardy hybrid grapevine and identified a QTL on chromosome 14 with a range of about 7 megabases. She thought this range was really too big to be useful. So they use a larger F1 population in a separate study for fine mapping using RAMP-seq haplotype markers. And we came to our target gene, which is it's more than 8 kilobases long. And we think it's responsible for phylloxera gall formation through gallic acid biosynthesis. And it has a very long name, which I will try to pronounce now. It's called bifunctional 3-dehydroconate dehydratase / shikimate dehydrogenase. We just call this the shikimate gene because it's just much easier than saying that whole thing all the time. And we call it the shikimate gene because it's part of the shikimate pathway, which is just a biosynthesis pathway for protein production, secondary metabolites, which are good for plant defense, growth, and development. From Dr. Lugian's previous research, he kind of hypothesized that the resistance to phylloxera is found at a single locus too. So we're investigating this gene sequence using PCR and Sanger sequencing, and we want to investigate sequence polymorphisms through these methods. So we're sequencing our target gene, and we're aligning to reference genomes in Vitis vinifera and Pinot Noir. And we want to see if a polymorphism is responsible for resistance in cold hardy hybrids. Essentially, we want to look for any key differences between our gene and our clonal cultivar and our reference genomes. The plant material that we're using for our grapevine was propagated asexually through cane cuttings. Our grapevine cultivar is called MN1264, which is just a cold hardy Vitis hybrid. And we down here, you can kind of see the how we go from our cane cuttings to budding to more vegetative tissue to finally we get our fully lignified grapevines. So direct delivery again, it's essentially the same as what we did in apple. We take our whole plant, we prune the top, we designate our nodes, we defoliate and wound, and then we inject into our nodes. The only difference is that this is how we cut our grape nodes. We kind of just aggressively just slice down the whole thing and try to cut as much as we can out of it. And this is just how we proceed with our direct delivery. So we also want to check our gene edit efficiency in our grapevine. So again, we want to look for a complete knockout. And since our candidate gene is not a reporter gene, it will not show us a very obvious phenotype. We start with our Luciferase assay. So similarly, we treat our leaves with luciferin, and then we observe the presence of bioluminescence under UV light. If we see bioluminescence in these edited leaves, we can then proceed to PCR or design primers just outside of our gene, our target gene. We then run a standard PCR, run gel electrophoresis to check for bands. If we do not have any bands, we can probably conclude that the gene was in fact knocked out. But if we do get any weird banding at all or see something that we're not expecting, we can then proceed to Sanger sequencing steps. So some future directions for our grapevine. So with transformed plants, we can then propagate our cuttings from edited shoots. We can treat with phylloxera to see if resistance was in fact lost. And then we can perform knockouts on commercially viable varieties potentially in the future. And of course, we want to keep fine tuning our procedure efficiency. So currently where I'm at in my project is our grape phylloxera gene is about 95% sequenced and aligned to a Vitis vinifera reference genome. We performed several experiments in both apple, seedlings, and grapevine, all at various degrees of stages. So we have some where I just started a couple of weeks ago, so we're still culling all the growth off. Some of them I just recently stopped culling the growth and let the injection nodes start to grow out. And in others, they've grown out for a while, and we're just waiting on me to develop protocol for our Luciferase assay so I can fully investigate if our knockouts were in fact done, which is very exciting. And of course, we're thinking about more experiments to try. And down here, you can kind of see some hopefully edited plants. So the first one is just a small grapevine that we have. The second picture here is of our apple seedling. We can kind of see how the node has like this weird overexpression from our leaves, kind of, which the developmental regulator is responsible for, and it kind of resulted in, as you can see in this last picture, these leaves and these shoots to kind of just explode out of our nodes. And with that, some acknowledgements. I would like to thank my advisors, Matt and Jim. I'd like to thank Dr. Liu Yin and Michael Maher for making this project possible. My lab group, the Fruit Group, you guys know who you are, for always helping me. Those in the Voitas lab for helping me with my molecular methods and our constructs. My undergraduates that occasionally help me, Quinn and Gina, thank you for all the help. And I would like to thank the APS cohort in general, just for all the support. And with that, are there any questions? Yes, James.
What's the process of identifying the target gene, and how difficult is that process?
So James asked, what's the process of identifying our target gene, and what was the second part?
What's the difficulty?
And what's the difficulty? Do you mean in edited plants, or like?
If you want to manipulate a gene within a plant, how do you know what gene that you're targeting?
Oh, so you know what gene you're targeting by. So this comes from when you design your modular backbone. It's all based on your guide RNA. You go into like Benchling or Genius or some program where you can design your guides, and then you design this kind of like complementary to your actual gene sequence. What I like to think of it as is kind of like, it's kind of similar to PCR primers, where you have like your guide and it just like knows where to like attach from there, and then you like kind of cut off.
Does the DNA has to be sequenced first? Is that so you know what?
Well, it doesn't have to be sequenced, but if you know, like for example, we found this gene in our from our QTL studies, and to actually design our guide RNA, we just used a Vitis vinifera genome reference, and we essentially just took the sequence out from that, and we designed our guide based off of the Vitis reference genome, and that's how we designed our guide from that. And I mean, they're similar enough. We blasted this, and they were about like 99.9% like similar between species, so we're not too worried about the guide being a good fit. It should work.
Any other questions in the room? So we do have one on Zoom. So the question is, how much effect of cultivar is there on efficacy of direct delivery?
Oh, right. So the question was, what's the effect of direct delivery on different cultivars, I guess? And the answer to that is, we don't know. I mean, it differs between species. There's a lot of differences that can kind of affect your guides and like your cast efficiencies. For example, in Grapevine, if you have higher GC contents of your guide RNA sequence, this actually improves your efficiency. So if you have your GC contents upwards of 50, you'll have way higher efficiency than you would at GC contents of 30. But it also kind of comes down to just the physical properties of the plants. So these are woody plants. And let me tell you, trying to inject these woody plants that are very hard and lignified with a very thin 30 gauge needle is a complete nightmare. If my undergrad is watching, she can definitely attest to this. The plant material is actually so hard that you have to worry about bending needles all the time. And then if you do actually manage to get your needle into the plant, you kind of can't even press the needle down. So it kind of creates like this pressure system, and like you just get liquid kind of like flying all over the place, like on the benches. You have to later clean up. But there's also an extent to where you get about, I don't even know, like 0.1 mil of cell solution into the plant. And then after that, the plant won't take anymore. It'll just kind of leak out of it. And we kind of just take this as a win if anything, if any of these cells can get into this plant and replicate at all. So going back to your question, it definitely depends on the physical properties of the plant. I guess internally, like sequence-wise and genetics-wise, we don't really know yet. We know CRISPR works very well in some plants and it doesn't in others. So hopefully I answered your question.
So there's another question in the chat. So what do you think the consumer acceptability would be for a fruit developed using these methods?
Is that from Seth?
Yes.
Yeah, okay. I'm sorry, what was the question again?
So, what do you think the consumer acceptability would be for a fruit developed using these methods?
So as far as consumer acceptability goes, my hopes are not very high, but yeah. I mean, people see CRISPR, they see like the acronym, they get kind of scared. They know gene editing has bad connotations, but I would eat like Arctic apple, like I would eat those if I saw that in a store. I think that's pretty cool. I'm not really afraid of it. But as acceptability goes, yeah, my hopes aren't very high. Hopefully we can educate people in the future to kind of show that this isn't that bad and perform more experiments and like say like this isn't that bad, or maybe make the USDA kind of like provisionary process more like available to people. Be like, okay, like we test this out like in mice and blah blah blah, mice, like dogs and like monkeys or something, so like it's therefore safe for humans. But there are actually a couple gene edited products on the market. One of them is actually a mushroom. They actually developed a mushroom that doesn't brown that you should be able to buy in stores soon, I've heard. And this is actually past USDA processes. Yes, Hannah.
Would this be fruits coming from these gene edited plants fall under like the sphere of GMOs? But if the genetic material is coming from the same species of plant, then do you have to sort of disclose that?
What do you mean by that last part?
Well, and he said that if you have genetic material coming from the same species of plants, you don't necessarily have to disclose.
Oh, right.
And it doesn't fall underneath the genetically modified technically because the genetic material is all still within them.
Right. Yeah. So the definition kind of changes between countries. Oh, right. So Hannah asked how I'm sorry, what was your question again? It's like
this would be necessarily considered genetically modified
oh, right.
organism or a GMO on the market.
Okay, so Hannah asks if using these processes, if this would be considered a GMO on the market. And yes, it will. It would be considered a GMO both in Europe and in the US by USDA standards. Actually, gene silencing, like the Arctic apple, is not considered GMO because you're not actually taking anything out or adding anything into it. You're kind of just, like silencing it. So that's technically not a GMO. But when you perform like a knockout, then that goes more into the realm of it becoming a GMO. That answer your question?
So there's no more questions in the chat right now. Are there any more in the room?
So, for the Phylloxera resistance knockout, it's just evidence that that is the resistance gene, right? It would actually make it susceptible, right? And so it's the idea is what that would be, kind of in an applied sense doing is just showing that that is what it's giving the resistance.
So Michael asks if taking out our candidate gene for Phylloxera will make our plants susceptible, and yes, it will. And yeah, that's not really the point of this study or of our experiments. That's kind of something that we can then test later on in the future. But we first have to see if we can actually take out this candidate gene in the first place using these methods, and doing this in soil grown environments instead of tissue culture. And then if we do take this out, then we can then go further into seeing, further testing the gene function of our candidate gene. But yeah, so it will make it susceptible by taking this gene out, but this isn't the point of our study. It's just to see if we can actually take these methods and apply them to something that could be used in, commercial ways. But yeah, in the future, I would love to test that. Depending on when I can finish up these preliminary experiments and hopefully get knockouts, I could probably just do this for fun. Just try to repropagate them and see, and then treat them with Phylloxera like on the side and see if we actually can use these methods to test for gene functions in the future. Answer your question.
Okay, so there's a couple of questions in the chat now. So the first one is, are there any potential genes known in apple that are breeding relevance, i.e. associated with important traits that could be targeted with these methods?
From our breeding program, I know that we do have a couple candidate genes floating around. As for if we could actually take these genes out and do more applied studies of them, I'm not really sure. Apple genetics is pretty convoluted. Maybe if the genes were like monogenic, we could probably do that. But if they're really polygenic genes that kind of code for a lot of different other functions, it might not be such a good idea. Because the common problem using CRISPR-Cas is you can get off-target knockouts kind of like downstream, and that can cause problems in the future. But yeah, I mean, I'm sure with further testing, we can take these candidate genes and try to do more with them and try to take them out, or maybe insert bases into them and see.
So the next question is from Lou. So she asked, given there are a few candidate genes in the Phylloxera fine map region, do you think it would be good to fine map even more? Or are you just testing out shikimate gene and then if not, try other genes?
So Lou is asking if we want to continue to fine map our region and see if there are any other genes beyond our shikimate gene. That's a good question. I'm not really sure if we do get a knockout with our Phylloxera, our target gene that we have currently, I don't think we really need for our purposes to fine map any further, but it would be very interesting to keep fine mapping because there are actually a couple other candidate genes. Just from what I remember looking at her publication, some of the graphs of the log scores, I think they found a total of like 153 candidate genes and kind of narrowed it down to a couple. But from what I remember is some of them had very high log scores. I remember this one was like, I mean, just like ridiculously big, but there was a couple other ones that were also very large. And I would actually personally wouldn't mind looking into those genes as well and looking beyond our shikimate gene, or maybe even doing experiments where we design guides and we cut out multiple candidate genes for this and kind of investigate that way. But yeah, I think fine mapping probably would be a good idea for a future experiment, not necessarily for ours though.
Any more questions in the room? I don't see any more on Zoom. Okay.
All right, well, thank you everyone for listening to my talk. I hope it was informative and not too boring. So thank you.
Who said thank you?
Yes.